Yes, after sharing my screen, it's my great pleasure too to welcome you to this first webinar of our Pandemic and Disaster Preparedness Center, in which we present one of the first topics that is an example of the type of projects that the PDPC will be developing in the years to come. But before that, I take the opportunity to briefly introduce our center to those of you that are not yet familiar with it. This is an initiative with a four-person leadership team, the names you see here, bringing together expertise from the biomedical sciences, technical sciences, social sciences, and public health and medical sciences, under a joint umbrella. Now, the question, of course, is the big why. Well, what we have now, of course, all become familiar with is what a pandemic looks like. And the issue that we address and want to address is that this is a true warning of things to come, because there has been consensus in the emerging disease field that we need to prepare for more and bigger outbreaks, including pandemics, because of the way the world is developing. That has to do with demographics, with our impact on the environment through large-scale livestock farming, the global mobility, and aspects and consequences of climate change and the impact on the planet. Now, in discussing this with colleagues online here, we realized that in the climate preparedness work and climate impact preparedness agenda, there are big parallels in the lines of thinking, but also the types of research that needs to be developed. This is an example from impact work on impact of hurricanes, example of the type of work that our colleagues from Delft are working on. And there are quite impressive parallels if you dig down into the scientific questions behind this. And the third element that often has been ignored, which has become very, very clear in this pandemic, is just how important societal challenges an epidemic like this, but also other types of disaster have, and that we need integrated responses, but also integrated research agenda in this arena. So that's really the ambition of our center, is to study the complexities of pandemics and disasters in combination through convergence of disciplines and taking a whole of system view. So looking at the pathogen side, but also the society side, technology, and the world around it. And we are aiming to develop this with a long-term agenda and stable long-term funding. So in order to do that, we have over the past year been working on a joint research agenda, which has three pillars. One is pandemic preparedness focused, one is water and climate related disaster focused, and the third one is focusing on societal preparedness. And for all of these, we are developing joint work, particularly through crossover studies, where we bring together teams around really challenging questions in this arena. The work can be from very basic to translational, and we of course also see the joint thinking flow and help prepare better for future disasters, whether they are from infection side or other disasters. And the societal preparedness, of course, very much focuses on the impact of on society. This is not something we do in isolation. What we aim for is to really not address everything there is to address. There are many people that are working in this space, but to really pull together tough challenges where we feel that nationally there is a very strong focus and that there's a good opportunity for international collaboration with some examples of already existing networks here. So in order to do that, we have a way of working, and that is through joint interdisciplinary projects. And the first run of that has been through 5 frontrunner projects that have been given budget with a task to come up with a joint proposal for addressing a key knowledge gap. And in the next few webinars, we will present them one by one. The frontrunner projects are interdisciplinary, challenging questions, and linking together those national and international partnerships that we want to see. And here is the 5 topics, and today the focus is on airborne. And why that? Well, we have seen, and you all have become familiar, I think, with the hot debate in the general public, in talk shows, among academics, in public health, on how exactly does this virus transmit, and how do we use that knowledge to design optimal control measures. And it's clear that understanding transmission is a very complex puzzle that involves the virus by itself. It involves people that have a certain health background. There is the interactions between them that involves air, but also surfaces. There's behavioral aspects, and there is facets of the built environment. And that is why there's a lot of outstanding questions, and our first frontrunner project really is aiming to address some key knowledge gaps there. So with that, let me hand over and introduce the chair of the first frontrunner project, who is Professor Ron Fouché from Erasmus Medical Center, and who has, of course, a long history in this, trying to understand how epidemics and pandemics of influenza arise. Ron, over to you, please.
Thank you very much, Marion, for the introduction. It's a great pleasure to introduce our proposal on predicting, measuring, and quantifying airborne virus transmission. I think it's clear to everybody now, due to the COVID pandemic, that non-pharmaceutical interventions are really key to mitigate the impact of a pandemic early on, when vaccines and drugs are not yet available, but also remain key throughout. And I'm showing some of the pictograms here on the slide that indicate some of these non-pharmaceutical interventions. Now, many of these mitigation strategies target the mode of virus transmission, and respiratory viruses are transmitted via direct contact, indirect contact, or via droplets in the air, large droplets or small droplets. But what is key is that the relative contribution of each of these transmission routes, so via contact or via the air, is often unknown, and actually remains unknown, as Marion already indicated, even 2 years into the COVID pandemic. And because some of these mitigation strategies can be rather disruptive, it is difficult, but also important, to show the relative contribution of modes of transmission and the way we mitigate the risks of these transmission, for instance, through physical distancing, through hand washing, through the wearing of face masks, through the ventilation of rooms. There's been discussion about this all along and continuing. And so measuring the potential contribution of airborne virus transmission and the relative contribution compared to contact transmission is absolutely key in pandemics, and may actually also be very useful to know in epidemics. Next slide, please. And so the complexity of transmission is shown in this slide. So there are 4 major routes of virus transmission when we talk about respiratory viruses: via direct contact, such as handshaking or hugging; indirect contact via your cell phones or doorknobs or table surfaces; and then there's the transmission potential via the air through large or small droplets. Next slide, please. And each of these transmission modes is very complex and will be targeted in our research program. When we look at the host aspect, so the person that is infected, there's incredible variation between different viruses in the amount of virus that is being shed, the site from which this is shed, the upper airways versus the lower airways, for instance, the duration of virus shedding, whether these viruses are expelled as single particles or as large aggregates that could only fit in a large droplet, and whether the virus is actually triggering sneezing or coughing reactions that would promote the expelling of viruses into the air. Also, on the receiving end in the host, there are many variables that impact the mode of transmission, such as the infectious virus dose, whether one particle is enough to cause an infection, or maybe, like has been suggested for COVID, 1,000 virus particles may be needed sometimes to infect, what is the infection site, whether the virus needs to be deposited in your upper airways, in the nose, or in your lower airways, in the lungs, whether there's already some cross immunity, and whether there might actually be some differences in genetic or otherwise susceptibility of people. But today we will mostly be talking about this transmission in the air, and that brings another number of complex issues, such as how stable will this virus be in droplets? What is the size of the virus particles, and would they only fit in small droplets or only in larger droplets as aggregates? How is this affected, the infectivity of the virus affected by temperature and humidity? And this temperature and humidity can also affect the droplet size. Droplets can grow in high humidity and can shrink in low humidity conditions. And how do changing salt and pH conditions affect the stability? Next, please. And so the aim of this proposal is to develop methods to predict and quantify the airborne transmission potential of viruses. And this work is split in 4 work packages that we will very briefly now introduce to you by 4 speakers. The first speaker will be Sander Herfst, who will be talking about work package 1, where we will optimize samplers to sample air, to sample particles in air, to test virus survival, but to also evaluate decontamination options. And then in work package 2, we will explain not how to optimize the samplers, but to how to optimize the sampling. So where to sample, how to best sample, and how to assess the exposure to viruses in the environment. The third work package will be presented, or the second work package will be presented by Inge Wouters. The third will be presented by Peter Frey, who is going to work on the host aspects. So how much virus is being expelled by an infected individual, what is the infectivity, from which side is this coming, is this virus singular, is it, and Peter will be introducing this work where we will be sampling in hospitals and doing laboratory studies into these aspects. And then the last speaker will be Philomena Bluisen, who will speak about the impact of airflows in indoor environment and how droplet sizes affect transmission in indoor environments, and also what are options for ventilation and other mitigation strategies. Next slide, please. And so we have assembled a very multidisciplinary team, as already indicated, from 4 large hospitals, universities in the Netherlands. And we are advised and supervised by Marion Koopmans and Aya Schreier in our work package. Next, please. By the way, all those people will be online at the end of this session to answer your questions. Just for reasons of transparency, we are listing here our site activities. All of the speakers today, of course, have other grants from various sources, as you can see here. All of those speakers also have various roles, advisory roles, largely unpaid, which may be seen as a conflict of interest to some of you. All of us have roles as editors or reviewers. And so just for matters of transparency, we highlight those potential conflicts of interest here for all of the speakers up front. And without further ado, I would like now to move to the first work package, which will be presented by Sander Hegst. Sander, take it away.
Yes, thank you very much, Ron, for this introduction. Next slide, please. So in this first work package, we will use experimental laboratory systems to mimic the transmission of viruses via the air between individuals. Next, please. So we will use aerosolizers or spray systems to mimic the expulsion of viruses in the air by an individual. The airborne phase in the middle will subsequently be mimicked by an in-house developed 3D printed aerosol chamber in blue, which I will explain more in the next slide. And subsequently, on the right side, so-called air samplers will be used to collect the viruses from the air to mimic the inhalation of viruses from the air by an individual. Next, please. So this is our experimental setup that I'm going to explain a little bit. So number one is an aerosolizer, or a nebulizer, and this is used to artificially generate virus-containing droplets in this aerosol tube. Number two is a ventilator. So this ventilator makes sure that the virus-containing aerosols and droplets are circulated in the tube for a predefined time period. After that time period, so we open the valve, the red valve in the middle of the picture, if you can see it, and the viruses will go to number three, which is the mixing chamber. Virus-containing droplets are very small. So first we mix the virus-containing droplets with mist generated by a mist maker, number four. And that mixing of these virus-containing droplets and the big mist will enlarge the size of the droplets. And as a result, we can more easily collect them with an air sampler. So that's point number 5. So these air samplers are used to collect the viruses from the system, after which we can analyze the samples and study the infectivity of those viruses. Next, please. So, however, the collection of infectious viruses from the air is still very challenging, and therefore we will first use this innovative laboratory setup to improve and optimize air samplers to accurately quantify viruses and respiratory droplets in the air. The knowledge that we will obtain on the air sampler improvement will then directly also be applied in the other work packages. So secondly, we will use, as Ron has already explained a bit, we will use this laboratory setup to perform some more fundamental studies on how the survival of viruses in the air, so the ability of the virus to remain infectious, how that is affected by, for example, the size of the virus-containing droplets or aerosols that are exhaled by an individual. Furthermore, we will study how the climatological conditions to which the viruses in the air are exposed, such as temperature, humidity, and UV, how they affect the stability of the viruses in the air. The temperature and the humidity of the air may also lead to evaporation of the virus-containing droplets, which result in a change of the pH and salt conditions and protein concentrations in the droplets, which may again also affect the virus survival in the air. So this will be studied as well. And these experimental results on virus survival in the air will subsequently be synthesized into a modeling framework that will be developed in work package 4, so we can estimate the transmission efficiency in relation to all these studied factors. And lastly, the setups will also be used to explore novel air decontamination strategies using UV LED and/or electrostatic inactivation using charged filters, which we can just build into this experimental setup. And these novel air decontamination strategies can potentially then be applied at a large scale in, for example, ventilation systems of buildings. So it was all very brief, but I hope that I have clarified our plans and our ambitions in this work package. And then, well, I will give the floor to Inge Wouters.
Yeah, okay, thank you. Next slide, please. So the aim of the work package 2 is to actually improve ways to measure the exposure in real life settings. And the picture on the left-hand side really depicts our main question. So what are existing viral concentration in real life environments? What Sander just explained is laboratory settings, but in real life, sometimes things are different. And ultimately, we would like to know associations between the exposure levels in the real life environment in relation to transmissions. However, before we can do this, we first need to be able to measure viral air concentrations. And what we have known from the past 2 years, it is quite difficult to do that in these settings. So the aim of this current work package is to see whether we can compare methods that are existing and available to apply methods that are being optimized in work package number 1, but also tuning of just existing methods where small differences may help to enable to measure the viral content that is able to replicate. So the viable virus. An example of that is, for instance, an existing method in the picture on the right-hand side, where we can measure in different size fractions within one sampler, but by adding fluids during the sampling collection, it seems that the virus viability can be increased, which means that we are able to measure viable virus in environments where we previously weren't able to do so. Next slide, please. Another thing is important when thinking about how to measure exposures, how to set up these studies, is that the exposure is not constant. So there's exposure dynamics. So what you can see in the right-hand side, the picture on the right-hand side, which is a hypothetical example of things that may occur. You see peaks of exposure, but you don't know when these peaks do occur. So we can imagine that if people are coughing, you see a peak in exposure, but it can also be that host differences may explain why some people do have higher responses than other responses. And we don't know at forehand what is the case in a situation in the real life. We take that into account, or we can take that into account by changing our sampling procedure. So for instance, taking longer duration samples will make sure that some of the peaks will be captured during sampling, and that this may increase the capabilities to really measure viral concentrations into the air. We don't know how these changes, exposure dynamics are changing over time, and that may be host dependent, but may also depend on different virus variations. And that is something that we would like to include in this work package. And that will help us to drive exposure assessment strategies for virus in general, but also combining it with the information from the other work packages to, in the end, increase the knowledge on how to model the potential of viruses and the pandemics from that. I would like to leave it for that. Also very short, and happy to answer some questions later on.
Thank you. So I think it's now my turn.
Yeah.
Inge is still on there. Can I start? Not quite sure.
Yes, please do. Go ahead.
All right. My name is Peter Fry. I will tell you what we're doing. Can I have the next slide, please? So as you know, we're trying to predict what will happen when the next pandemic virus hits us. And as you know, those pandemic viruses do not originate at random. So most of them originate from virus families that are already widely known, and they just have waiting to cross the species barrier. And of those virus families, we have well-known archetype viruses, viruses that kind of sums up what's going on there. And you probably know them, you listeners. So influenza, RSV, and they are every year again, they cause outbreaks in kids because they're treatment naive. For children, actually, every young children, every virus is a pandemic virus. And what we're trying to do is see what's happening in those kids. As surprisingly, though, all those viruses are very well known, we know very little on how they actually infect us. So what we're doing, we go to infected kids, and then we're measuring in exhaled breaths virus or droplet size where the virus is encapsulated. As Inge already explained, by deducting from those the sizes of those particles, we could try and understand how the virus spreads through the air. But we also want to do additional stuff. Next slide, please. apart from knowing the amount of virus that the kids are shedding and which fraction or which sizes of virus they're shedding, which is not necessarily the same for all the virus families, we also want to know whether the virus is replication competent when we catch it. And I think for easy sake, you can say whether the virus is alive. And I think biological, it's not totally sound to say it like that, but it is what we're trying to do. And how that works, you can see here. So on the left-hand side, you see me looking through a microscope at a cell layer, and then we add, so nice pristine cells, we add some virus containing fluid to that, for instance, what we captured, and then you see that the virus actually makes a cell ill. You see destruction in the cell layer, and you can color that. Now, this gives us enormous amounts of data and ideas on how those viruses spread to the virus and whether they maintain to be live. But we also encountered some other things. We also understood that despite all the difficulties that Inge was already talking about in Sander, that viruses have to survive to go to the air, there's also the host. Just recently in the COVID times, we especially my colleague Jeroen van Kampen showed that despite the fact that there's a lot of viruses, COVID viruses present in infected individuals, sometimes we can no longer culture the virus, that it's no longer replication competent. And that has to do with the amount of, a certain amount of antibodies that for some reason interact with the viability of the virus. Next slide. So if you add antibodies, you see that the culturability is no longer there. Those things we'll be studying in peacetime. When we're not sampling the kids, in peacetime we'll be busy trying to understand how that works. If you think about it, it's almost unbelievable that a virus actually manages to infect us, but well, pandemic showed that clearly goes quite well. But we're very interested in how those host things and predict whether a virus is viable. Nice collateral damages of these studies will probably be that we have a better approach to how isolate people and how to done and done off personal protection, because that's important for hospital stays and for the amount of hospital people that we can allow in hospital, but also something that's a bit neglected, the amount of waste that we create every day, every year, especially in the pediatrics when we have those huge outbreaks. So I'll now give it over to Philo.
You are muted, Philo.
Philo, yeah.
You hear me now?
Yes.
Yes. Okay. Thank you, Peter. Before I go into the specifics of work package 4, just some background information. We already heard a lot about that an exhaled droplet, the so-called respiratory droplets, from an infectious person can contain the coronavirus. Now, these exhaled droplets have a wide range of sizes, and most are in the range of sub-micrometers to a few microns, of which the larger droplets actually quickly fall to the ground when they are exhaled, while the smaller droplets can rise and can linger in the air. Next, please. How long and how far a droplet can stay or travel in the air depends, therefore, on the droplet size, the contents, and on the local airflow conditions, like air velocity, temperature, and humidity, the same conditions that actually also influence the viability of the virus in that droplet. Next, please. Now, second, to decrease the risk of transmission of the virus indoors, the use of proper ventilation measures have been recommended. We all have heard that the last couple of months. But what is exactly proper ventilation? This is something we would like to investigate. Proper ventilation means, first of all, to provide sufficient and effective ventilation. Ventilation that ensures the supply of clean air and exhausts the infected air from the breathing zones of each individual person, preferable without passing through the breathing zones of other persons and without recirculation of air. If general ventilation seems not enough, then you can always use air cleaning devices. Now, ventilation can be established by just opening a window. We call that natural ventilation. or by using a mechanical ventilation system, varying from only exhaust to very advanced air conditioning systems that supply and exhaust the air. Also, different ventilation principles can be selected, such as mixing ventilation. The air is then mixed completely through the room. Displacement ventilation, the air is going from one side to the other side of the room. Cross ventilation, same principle. And personal ventilation, where you exhaust the pollutants directly in the neighborhood of the person. Now, all those different regimes, as we call them, can create different airflow conditions. Next. Now, how the virus containing droplets can be properly ventilated or cleaned depends therefore not only on the amount of ventilation, but also on the way we ventilate or clean the air in a space, creating all these different airflow distributions. Next. Now, therefore, in work package 4, the intention is to characterize the distribution of pathogens or viruses and pollutants indoors under different ventilation regimes by making use of the outcome of the previous work packages, so the sampling in the air, but also the infectiousness under different conditions. We will use advanced open source computational fluid dynamics tools to make this model, this prediction model. And we use experimental studies, both in the lab under controlled conditions, as well as in the field, using different living labs, and that's the attention. And using different techniques. So we'll use chemical techniques, physical techniques, but also human assessment techniques. So the next, the anticipated outcome of work package 4 is a 3D model for the distribution of droplets and the impact of different ventilation regimes and environmental conditions on the transmission risk, including all the previous information. We will have cases in living labs, and we still have to decide which particular living labs we will use for that. And hopefully we will create a prototype of a ventilation system for a common indoor space in which we can test our model, but also to show how we can provide new types of ventilation for future spaces. Thank you. Back to Marion.
Yes, thank you very much. And thank you for that brief introduction. And as you can see, we now have a broader panel, and that is so who joined are several of the additional scientists that are involved in this multidisciplinary project. I would like to introduce Jerome Wishaupt. If you please just wave your hand so people know who you are, from Reinier the Graaf Hospital, who is a medical doctor pediatrician who is involved in the air sampling studies in children, where I briefly thought Peter Vrij said he was going to infect children. I just want to clarify that that's not that part what is going to happen, but the studies will happen in the settings where sick children are present.
Definitely, sorry for that.
Yes. For that. Just to clarify, we have Bas Kolen from TU Delft, who is an expert in risk modeling and has a very strong background in the disaster preparedness field. Additionally, online is Guoqi Zhang, and I'm sorry if I mispronounce your name, TU Delft from electronics, working on, among others, UV inactivation and other ways of thinking about inactivation. And Hanneke Gelderblom, yes, and Tess Homan, both from the Applied Physics Department, TU Eindhoven, and working on fundamental studies on particle physics. And then last but not least, Anja Schreier who is medical director of medical affairs of the Pandemic and Disaster Preparedness Center, and also with his interest and background in public health research. So thank you for that. So this was a somewhat lengthy introduction, but we want to make it clear how, as PDPC, we are addressing things so multidisciplinary, challenging. So with that, let me start by asking a first question, maybe starting with Bas Kolen. So given your modeling expertise, of course, what we have seen over the past 2 years is a lot of discussion about different elements of the airborne transmission. And here we see, well, hopefully a dream project where there's all these pieces of information together. I'm sure that must be a dream for a modeler, but I would be very interested in hearing how you would, which challenges you see ahead.
Thank you, Marion. And well, from the modeling point of view, of course, I would like to know whether how the virus spreads. Is it by contacts close by, and is it by contacts on a further distance, by the airborne part, and how far what is the range, and how infectious is the virus, and when I look at events, so where I want to apply these models and to make choices, how can we design such events or bars or etc. And so we have information about, and well, where do we have to put the tables, how many people are allowed, but also I think related to the work of Philo, how can we design well, the ventilation in such a way that, well, it flows not through the people, but it goes away from the people. So from the modeling point of view, I'm very interested in these parts, and also the certainty or uncertainty for the estimations we have, so we can make better estimations about, well, how effective measures are.
Okay. Yes, maybe a follow-up question for, I think both Inge Wouters and Philo. You show the complexity of the real life, which is always difficult if you try to research things. Philo, you showed these different ventilation types. There's of course very different ways rooms or buildings have been designed. How do you see the extrapolation of what the work that you do, the observations that you will have to other settings? What is what are your challenges there, or how do you see that move forward? Maybe Inge first.
Yeah, I think it's a good question, and I think we're really at the starting point to study this. So what you can do in our types of studies is that you do observational research, where you both capture the information on, you know, what you say, the setup of the rooms where you are sampling in, what people are around, those kind of things. And in combination with the information that Philo is getting from her models, you will be able to maybe draw up a general pattern that could be applied then further on in the risk modeling sites. So it's really a starting point at this point in time, and thus far we were just struggling to actually be able to sample viruses in the air at all. I think we really have improved that over the last few years, and I think now we are at this first step to be able to do it in more repeatedly and in different settings where you can actually look for those relationships.
And Philo, do you want to add some?
Yeah, I would like to add some. What we see in the studies that will be performed, for example, by Sander, he will look carefully in the lab what the effect is of different climatic conditions, like temperature, relative humidity, and possibly also airflow, air velocity, on the droplet with the virus to estimate the viability of the virus once it is exhaled. What we can do in our Sense Lab, this is a lab where we can change the ventilation, so we can create different ventilation regimes. We can also add, for example, personal ventilation, and then we can measure on the spot the different air velocities, temperatures, and relative humidities that are created. We can also measure the droplets, the sizes, the different sizes of the droplets. We can measure that. What we cannot measure is the virus there. And we cannot just play with a virus in our labs. That's something that Sander can do. And perhaps in combination with the techniques of Inge, she could also then in Sander lab measure actually both the virus and the droplet sizes for that. Now, having all that information, we can put that in the 3D model that we are creating. So we know the parameters that are important to turn on, to know what eventually the transmission risk is in a certain situation. So we can also translate that to another case, as you would say.
Okay, yes, I see a question in the chat. One of the suggested cases is airplanes. And the question is whether you are thinking of including that.
We have not discussed that yet. We were more thinking of a situation that we are all braving for at the moment, and that's a restaurant or a bar.
Yes, priorities, and then after that maybe flights, but
right.
I think it's a good suggestion. Ron, you have
maybe it is important to emphasize that this is really a research project to develop methods initially. And so I also see a question from Luc Koffeng in the chat, also implying many more studies in the field, in different people, in different locations. But of course, there are limits in what you can do in a project like this. I think the first step is to develop samplers, to develop sampling protocols, to evaluate them, and to see indeed some applied projects, such as looking at the built environment. But you cannot do everything at once, of course.
Yes, thank you, Ron. That's a good addition. And the ambition here is to really tackle some of the really tough challenges. One of them is, for instance, how to assess the questions that you're interested in for particles of different sizes. So maybe that's a question for the particle physicist from TU Delft, Tess or Hanneke. Do you want to pitch in?
Yes, I think we can say something about that. So in Eindhoven, we study a lot the physics of droplets, and a key challenge is there not only, you know, if you sneeze and you exhale a cloud of droplets, it's already a highly complex physical process how liquids break into pieces and how these different virus particles distribute amongst those different droplets. So you would want to size separate those drops. And then an issue is when you do so, you also influence the droplets because they evaporate during this process. They impact on the surfaces on which you want to collect them. And by that, you influence also the viability of the virus itself. So to do this in a non-intrusive way in which you keep the virus alive, as you could say, and to have a size resolution. So to be able to say in what droplet size is sitting what amount of virus is a very challenging part that we want to address in this first work package, basically. I don't know, Tess, do you have additions to that?
Yeah, no, it's exactly what you're saying. So it's not only keeping the virus alive, but also tracking what happens to the droplets while you measure them. Because if you're interested in the droplet sizes, then you need to know what happens with them in, for instance, the air samplers, so that you really know what droplet sizes you are looking at.
Yes, it sounds so easy, just measure infectivity, right? Sander, maybe a question for you, because at some point in the longer run, we maybe are going to rely on these new methods for really assessing a situation, which means that the detection limits and sensitivities of these tools are going to be crucial. Given all the complexities, what challenges do you see there? And particularly, what will the negative test results come to mean?
Yeah, so the first challenge that follows up basically what Hanneke and Tess just said as well. So we now collect the viruses with samplers. There's a whole range of samplers that we can use, but if we collect those viruses, the impact on those viruses by, we smack them on the wall or on a filter, and everything we do to collect them has such an impact on the infectivity. So you lose so much infectivity just by the collection method itself. And me as a virologist, I can of course buy those air samplers and then I can try them in the lab. But what is very nice about this collaboration that we are going to do here now with Hanneke and Tess, they can do the calculations. They can maybe come up with ways that we are more gentle for the virus particles, so that we in the end up with infectious virus, which is something that we can show or demonstrate in our lab, of course. And that's a big limitation now, is that a lot of air sampling is done at the moment, also with SARS, but because of these samplers being so harsh for the viruses, you can't culture them, but you can only show that there's viral RNA, so there's genomic material of the virus is present, but just the presence of that RNA does not say anything about the infectivity of the virus, of course. So just showing viral genome in the air doesn't mean that it's necessarily airborne, because you don't know if it's infectious. And I think that's the biggest challenge that we have. And if we can do that, then of course we need to scale up to larger volumes, etc. Yeah.
So maybe that's an interesting follow-up is from the observations working in near the animal farms. That of course, so Inge, your institute at least has been involved in that, and there we see that a big part of the problem seems to be dust and virus-laden dust. How do you separate dust transmission from aerosol transmission? That's a naive.
Well, I think it's a question of semantics. The question is also in an environment in person. So in the animal facilities, there's just a lot of dust. There's furs. There's feeding material. So what is in the air? There is a mixture of things that are being shed by the animals. It's the fur itself. It's everything that comes into the air. The same is, of course, true in another setting with just humans. We sit here. We also shed all different kinds of things from ourselves. We shed dust. We have dust that is that was on the floor, and that can be re-aerosolized. And also in there, you can imagine that if virus is being shed, and like for instance, the virus, the bigger particles or the bigger droplets may fall down on the ground, they can be re-aerosolized as well. So I think in at least in our epidemiological studies, we will not be able to really distinguish whether it's dust or not. But we will be able to say, okay, it is in the bigger part or it's in the smaller parts, and we will be able to state something, whether it's really in the air or not.
Okay, thank you. Yeah, it's just that I was struck by the huge amounts of virus found in the animal environment and the far lower amounts of virus.
Yeah, but I think in the animal environment, we really were in a setting where all it was in a mink farm, so the minks were really being ill. And the density of the population is also very high compared to settings in human settings. You never have that human density as compared to the animal farms. So it's really different settings.
Yeah, okay. So maybe moving to the host side. So Peter, you explained the planned studies. Yeah. Jerome, so you've been at the receiving end. Maybe tell us a little bit about what your expectations would be from a pediatrician or maybe a stakeholder perspective. What would you like to see developed that would help you in the setting where you work?
Well, that's a difficult question.
in our hospital, it is important for us to have informed consent from the parents at a very early stage, so we can include those children in an early stage of the infection. Children have a native immune system, so it's nice to look in children whether how the infection is spread, and it is important to have single room settings, because otherwise, yeah, you don't know what you measure in air with an air sampler. So, well, what I would like is that there are no more hospital acquired infections, but that would be a long way, I think.
So, well, I guess that starts with understanding patterns of transmission. Maybe Gucci Zhang has, I understand you're interested in decontamination.
Yes.
You hear this question, where would you go?
I think the challenge for the decontamination is the effectiveness and efficiency, because in the air, and this airborne can fly everywhere, and we would like to make sure that the amount that remain in the air will not be contagious, will not, let's see, get people sick. So this is challenging because I'm not sure the percentage, the ppb, and how many particles or virus will not be harmful. So this is a big challenge, effectiveness and efficiency, but also very important if we work on the indoor environment, does not matter to be in aircraft or cars, trains, restaurant, and the cost also very important. Yeah, so this should not be very expensive and also reliable, not only working for 1 month. Some traditional technology and not really have a long lifetime. So cost, long lifetime, efficiency, effectiveness, and finally we need to optimize this. So there are different technology methods, the UVC, UVA, and also electrostatics and so on. But we need to further develop the most suitable technology, and also to fit into the current air system in the buildings, for example.
Okay, well, so that's more distant future, I would say, hearing you. Maybe back to Peter. I'm intrigued by the question of what, of course, we have seen with SARS-CoV-2, how important it is to know the host's immune status. And we've seen that by the time patients come to the hospital, they often have already high levels of antibodies, and the viruses are difficult to culture. So that might imply that maybe in a hospital setting is not the best place to do these studies. What do you think about?
If this will be the adults, I'd say you're totally right, but these are not. So that's why we chose for the young kids. So especially for those archetype viruses, I think the pathogenesis is a bit different. It's acute infection, and of course it's pandemic, so they don't have a pre-existing immunity. This will be very hard to do in adult for the archetype viruses that we chosen to do. Considering the clinical picture and the quickness that those kids develop disease, especially for flu and paraflu, I would think that initially they will not have antibody response. Would take 4 to 6 days, I guess, before that to be robust. And based on what my clinical expertise, will catch them earlier. But as Jerome said, it all stands and falls with the quickness in which we can able to include them. So we set up this whole thing for RSV. We just right before the COVID pandemic. Or actually during the COVID, we were able to get some data out of it, and then things were stopped. But it will stand and fall with how quick we can do it. But we set up that infrastructure now. So if this, I totally agree, if this would be pre-experienced individuals who have previously had flu or RSV, this will probably not be easy to make fly. But since these are the very young ones, you're talking about 1 to 2 years of age, I think there will be less of interference, and we may be able to measure even that interference there as they are initially in need. Over.
Okay, thank you. Maybe near final question to Anja Schreier. You have been working, of course, a lot from the municipal health service perspective with your previous position at GGD Amsterdam. And you're now looking at longer term scenario planning. Do you, if listening to this, what is your thinking about the role of the expertise that is being generated here, the knowledge that's being generated in our forward thinking about pandemic planning?
Yes, thank you, Marion. I think from a public health perspective, it's going to be very interesting to see how what happens if we validate these methods in settings like nursing homes, like childcare settings, like primary schools. We've seen these questions during the current pandemic. We've seen we did have some questions that we couldn't answer yet. So I hope in the not too late future we're gonna be able to answer some of these questions. Also to see what happens in settings with relatively more healthy persons, elderly, youngsters. So I think that's going to be an interesting phase of the study that I'm looking forward to. And also to help us to prepare for future pandemics, as been said in the introductions, because if we can prove that if we change certain things in ventilation or in infection prevention methods, that's going to help us to be more prepared for the next pandemic.
Okay, I'm looking at the clock. We are approaching the hour. So that concludes our first PDPC webinar. Before handing back to Ron for final word, as the chair of this topic, let me also announce that we will have these webinars repeatedly. If you have signed up, you will be alerted to them, and the next topic planned is somewhere mid-February, which will be a brief further introduction for newcomers, but will have a focus on forward thinking and using scenario analysis to plan for future disasters and pandemics. And some of the methodologies that are being developed there. So if you're interested in there, please join in the next webinar. So with that, Ron, back to you for closing of the meeting.
Yes, I hope that we have entertained you all and explained the project in some detail. Of course, we need to be very short in just half an hour of explanation. I also would like to point out that this plan looks very ambitious and is extremely ambitious and could never be completed in isolation. We are all working in international, other international groups connected to this. I already saw some people online from our NIH project, like Don Milton, that we're working with. But I also know that the people in physics in Eindhoven, in Delft, have other connections to projects directly linked to this one. And so projects like this are going to work like a multiplier of research that really connects the different disciplines. And I'm really looking forward to get this project going. And hopefully with the next epidemics and pandemics in the years to come, we can actually answer the critical questions that are out there.
Thank you very much, and thank you all for the presentations and for the panel discussion. And we see you again next webinar.